I now wanted to bring to the stage Al George, who many of you know. Raise your hand if you've met Al George. Raise your hand if you want to. You should. Okay, so Al George has taken a huge role in our foundation recently in getting our science going at a completely different level. If you came to Chicago, you were lucky enough to have Al George and the team at Northwestern University and Lurie Children's host us. That was so great that we've kind of folded him in more, and he is really leading us on a terrific journey. So Al's going to talk about Science is Better Together, insights from the Research Roundtable, which we had yesterday, which was a whole day of high level what we just heard. Ladies and gentlemen, can you please welcome our friend Al George.
Thank you, Sarah. I'm happy to be here as a scientist as well as a physician studying rare diseases. There's nothing more inspiring than come to a meeting where the room is filled with parents and their children. And this is why I do this. And I hope that what I can tell you in the next few minutes will give you some hope that there's a lot of science going on KCNQ2. Yesterday was a full day of research talks. I'm not going to repeat them all in 20 minutes, but I'll give you the high points of what was discussed, and some of the material that was presented will be replicated for you during today's meeting. The goal of the Research Roundtable, by the way, was to get together a critical mass of scientists, clinicians, people from industry, people from families, to put together a critical mass of people to think about what do we know now about KCNQ2, what don't we know, and why is it important for us to know that, and how do we go forward for the next few years and solve some of these important gaps in the field. So the structure of the meeting yesterday was to highlight research in 3 domains. The first domain was basic research. How do we model this disease in cells, in animals, and what can we take away from what we're learning now? The second opportunity was to discuss how do we start to work smarter at building a clinical trial, a structured method to test new drugs, new medicines, and what are we trying to improve in the lives of children with KCNQ2 related disorders. And that was a big topic of conversation over the last year in our scientific advisory board meetings. How do we get to where we are now to understanding what are the endpoints of a clinical trial? What do we measure, and how do we prove to regulators like the FDA that something is working so they can put it on the market? The last area was what's happening in the world of therapeutic development. What are the new opportunities to develop new medicines, new genetic therapies, and where is that going, and how can that be directed toward KCNQ2? The last part of our activity yesterday was built around basically a think tank, getting people together to talk about what are the gaps in our knowledge right now, and then how do we then translate those opportunities for learning more into a roadmap forward. And this is hopefully going to be helpful to the foundation to prioritize specific work to be done in the short term, in the mid-term, and long term, and be more focused in those opportunities. So that's sort of the framework. Now, I have the hardest part. I'm going to probably in about 10 minutes try to summarize 8 hours of science for you. And I'm not going to talk like an auctioneer. And you're going to see a lot of slides, and you're not going to understand them, but they're placeholders for just me telling you sort of, here's what we talked about, here's some take-home messages, next slide. So hopefully this will work. First of all, this was the agenda, and I don't want to have you struggle to read this, but this was the first session where we talked about model systems. To study a human disease, you really can't do that adequately with patients. You have to replicate the disease biology in some simple system that can be used in a laboratory. cellular systems, mouse models, other animal models. And so our talks were just to touch on mouse models and what we get out of them, and then human neuron models and what we can learn from them. So the first couple of talks, one was given by Tasso Tsugunis from the University of Connecticut. He had this beautiful slide that showed kind of a state of where we are in having available genetically engineered mice that replicate all of the core, the spectrum of KCNQ2 related disorders, all the way from sort of the milder form to the more severe form. And this is really great because now we have the tools to look at the physiology and the pathology of this condition in a living organism. He also highlighted a number of key questions that I think are important to answer at this point. And so one is, we know a lot about where KCNQ2 is functional in the nervous system, but which of the cells in the brain are responsible for the major disease manifestations of KCNQ2? Also, is there sort of some core mechanisms that we can identify that ultimately could be targeted with therapies? Is the brain's constellation of proteins, which is referred to as the proteome, affected by abnormal function of KCNQ2? And then, of course, mouse models are one platform to test new therapies in, but not the only one. Then we moved on to a recorded talk by Dr. Laurent Villard, who also studies one of the more common recurrent KCNQ2 mutations in a mouse. And he presented a very interesting story of how carbamazepine, commonly used drug in this condition, which is a sodium channel blocking drug used to treat seizures, how this drug not only may control seizures, but may also affect other behavioral and neurodevelopmental features of the condition. So there may be more value than just turning off the seizures. And he presented some nice data on that. I won't replicate that. We moved away from mouse models to talk about human neuron models of KCNQ2. And this was led off by a talk from my colleague, Dina Simpkin from Northwestern, who has put together a large data set of 5 different KCNQ2 patient-derived stem cell models of this disease. And from that, she can create neurons, brain cells, functional brain cells in a dish from individual patients with KCNQ2. And from that, she's been learning about the electrical activity of those neurons using a sophisticated technique called microelectrode array recording, where you can record from hundreds of neurons at a time and learn about their behavior. And the data gets very sophisticated. Here is something that, don't try to even read this, but the data are very dense. This is the result of millions of measurements of neurons that's been distilled down into a few morsels of information. And I let you, if you want to read this, but I think the goal of this work is to try to understand how not individual neurons are working, but how they work in groups and networks of neurons, which is more representative of the whole brain. Also using this technology of stem cell derived neurons were 2 graduate students from the University of Antwerp, Nina Dirks and Nora Zonneken, who also presented elegant work on using the same technology. And they're learning also about how early in the cultures they see different effects than late in the cultures. And so there's an evolution of the disorder in the dish, and there's a lot of information from that. And perhaps there may be other therapeutic targets to consider at different stages in the disease. Now we moved on to talking about clinical endpoints and clinical trial readiness. And by the way, this is a topic that permeates all rare diseases. How do we position ourselves to be ready for a clinical trial with willing participants and clarity about what are the endpoints that we need to measure for regulatory approval? And this was started out by Mark Fitzgerald from Children's Hospital Philadelphia in Philadelphia here, where he kind of gave a nice framework for how do we address the challenge of developing clinical trials and designing those clinical trials for something like KCNQ2. This was followed by Stefan Oven from Paris, who really laid out the challenges of doing that and how you methodically pick clinical trial endpoints to be effective and give you a positive answer at the end. We then moved on to an important talk, which you will hear later today by Michelle Potashman from Biohaven, a company that's developing a new medicine for KCNQ2. And she talked about the idea of building what is called a disease concept model. What is the disease? What are the features of the disease? And which features are most important to the families affected by this condition? And this is a survey-based, some of you may have participated in this, and they're getting ready to publish this information. But she gave a glimpse of some of the findings. For example, what's shown on this slide is parent responses or caregiver responses as to what are the most impactful features of the disease. And time after time, what has come to the top has been things like communication and motor function, not seizures. So the classic way of doing a clinical trial in this disease would have been to measure seizures. Doesn't work. Need other measures for that. And so that led to the next talk by Matthew Mill, also recorded, but he reported out on a new clinical trial, a retrospective looking back clinical trial where they looked at hundreds of individuals with this condition and how their response to carbamazepine, this standard sodium channel blocking drug, how they responded in terms of seizure control and other features of their condition, including neurodevelopment. And remarkably, they found that, yes, sodium channel blockers, including carbamazepine, were the best drugs for controlling seizures. But importantly, there was also evidence that those treated with sodium channel blockers had improvements in their neurodevelopment. We've often thought of sodium channel blockers as largely drugs to control seizures, but maybe there's more benefits to it than that. And this study helps to sort of open the door for understanding that. Now, one of the challenges with defining clinical trial endpoints, things to measure the effectiveness of a new drug in a clinical trial, is in children with KCNQ2, some standard measures of development and behavior don't work because the children have levels of impairment that make it difficult, if not impossible, to complete the questionnaires and tests that are required to do those standard measures. And so there has been an effort, including the effort led by Gabi Conacher, called the Inchstone Project. We normally think of milestones in development, but they're talking about smaller increments in progress that they've coined the term Inchstone. And the Inchstone Project is a vast project not specific to any particular disease, but now happily includes KCNQ2. This is an opportunity for people to participate, for us to all together learn what are the measures that give us positive outputs from a new medicine that are measured not in miles, but in inches, small increments. This was followed up by Natasha Ludwig, one of the major participants in the Inchstone Project. She's a developmental neuropsychiatrist, psychologist, and she will be giving a presentation to you as well to tell you more about this. But she also highlighted sort of the roadmap of how do you build a new measure for measuring a neurobehavioral outcome. It's not easy, but sophisticated researchers like Natasha are getting it done. Now, the last segment was the last session, which we did at the end, was on advances in therapeutic development, new medicines, new small molecule medicines, new genetic medicines. And so this was led off by Andreas Brunkklaus, who you're going to hear from as well later today, and he talked about the advances in treating another rare developmental and epileptic encephalopathy called Dravet syndrome. not related to KCNQ2, but has some overlapping features. But that disease has been the target for many new drug development efforts, including one that's moving now into the phase 3 clinical trial, which is the last kind of pivotal point in getting a drug developed. And this is going to be testing an antisense oligonucleotide therapy, also known as an ASO, as Brooke mentioned. And this is what he will talk about and how that's being put forward in a clinical trial fashion. This is just the clinical trial. Now, traditional small molecule drugs, like you're all used to taking and giving to your children, still some opportunities to find new medicines that work in KCNQ2. But the process of developing a medicine takes a long time. If you start from scratch, it can take 10, 15, longer years to get a medicine to market. So one way to short circuit that process is to look at available medicines that may have already been tested in some other disease. Maybe they've been approved, maybe they've not been approved, but you can screen for your gene of interest or your target of interest to see if there's anything out there already. And this is called drug repurposing. Find something that's already available and repurpose it for your indication. And this was an approach highlighted by Maurizio Tagliatella from Italy, who called in last night from Europe to give us his presentation. And he reported on their efforts to identify existing small molecule drug-like drugs to find something that activates KCNQ2, similar to what the goal of using the drug retigabine was. And they found remarkably, in a screen of more than 5,600 compounds, they found one that was a compound that had been developed by Johnson & Johnson for schizophrenia, but had then been shelved for unclear reasons. And they found that this drug activates KCNQ2 and could be a potential therapeutic weapon in this disease. Moving on, we heard from a couple of companies that were gracious enough to share some of the work they were doing. One was the company Curalis, who has a novel chemistry and a novel molecule that has also an activator of the KCNQ2 potassium channel, and they're moving that into early phase development to move forward in a couple of different indications, possibly including KCNQ2 epilepsy, and they came to the meeting to learn more about the disease. Another presentation from a company that you're going to hear about later today called Asymptote Genetic Medicine has a very clever idea, which is to, in the case of KCNQ2, get rid of the gene because it's causing problems, but then provide a fresh copy of the gene to the brain. Now, this idea of gene therapy has been around for decades. The challenge is, how do you tune the amount of the gene that gets in and gets turned on? That's the big challenge. Well, Asymptote has got a strategy now that they can do that very precisely, and you'll hear more about that later today from Acacia, from their company. Lastly, from a company perspective, we heard from, and I'll get his name wrong, Yuri, can't remember his last name now, I apologize, from a company called Camp4, and they have developed another technology that could be potentially used in KCNQ2, and this uses small DNA molecules, antisense oligonucleotides, to interrupt regulatory molecules that may control the expression level of the gene of interest. Lastly, we heard about a new effort to present the data that we have on KCNQ2, both from the clinical and genetic perspectives and maybe research findings, in one place, a one-stop shop for all things KCNQ2. It's a dream that will come true because of the efforts of Dennis Lal from University of Texas, Houston, and his team that builds portals to present information about a single gene from all perspectives. And they've just started to build one for KCNQ2. And this is just a screenshot of the pilot phase of that project. To get the data in there, he is eliciting surveys for caregivers and families parents and caregivers to input data about their children's condition. And I think that's just step one. He's going to be aggregating all of the genetic data that's out there in the literature and available and presented in a easy to understand format. This is a portal of information. It's going to be valuable to families that want to learn more about the condition. It's going to be very valuable for researchers and clinicians that want to see what the state of the knowledge is. And so we're very happy that he's doing that, and that's going to really, I think, accelerate research in the field. Now, the last activity that we did, I'm almost finished, is we wanted to get these minds together to identify, think collectively about where are the gaps in our knowledge about KCNQ2, and what are the opportunities to fill those gaps, and which ones should be prioritized in the near term, in the longer term. And so we decided that we would create a workshop-like atmosphere, bringing everybody together, sitting at tables just like the ones you're sitting at now, and to have these discussions. We did this in 2 phases. The first phase, we broke everybody up into groups including family members at one table, researchers in another table, clinicians at another table, and people from industry in another table. And we had them collectively think about what are the gaps in this field in their domain, from the family perspective, from a clinical perspective, et cetera. Then we had them all stand up and tell the group what they came up with, and we carefully annotated all of that on these big sticky notes and put them up on the wall so we could think about them a little more. So once we identified the gaps, we looked for the common themes across groups. Did more than one group come up with the same idea? Then that clearly is a high priority gap that we should attack. The second phase, and here perhaps are the gaps that we came up with, not in any particular order. The second phase of this exercise, we broke up into more random assorted groups, not just clinicians here and families there, but we mixed everybody together, and then we said, okay, your table gets to tackle this gap. How do we attack the need for a patient registry and the need for a natural history study, for example? And then tell us what you think is the highest priority to get done in the next 18 months, the urgency, what's something that should be accomplished by 3 years, and then beyond 3 years. And I hope, once we distill all this down, we will be able to hand this to the leadership of the foundation as a guide to creating a directed strategic plan for the foundation. Where do we put our resources? Where do we put the emphasis? And how do we as a group tackle these and fill these important gaps in knowledge? So here's just some, this is an example of some of the things that we came up with that should be prioritized, or at least considered for prioritization for the next 18 months. And you can read them. One is to develop a registry of everybody with this disease and standardize terms so that when we're talking about this, it aligns. There's also an opportunity to build more of a natural history study. Why that's important should be clear from other speakers today, but in a rare disease, knowing the trajectory of the disease in the absence of new therapies is very valuable for regulatory purposes. And you can read some of the other ones there. I think an important one is defining clinical endpoints for a trial that are not seizures, something beyond seizures. And then there are some that are more on the research side, understanding what's available in terms of research tools, who has which mouse models, who has which stem cell models, and how can those be aggregated, that information aggregated into one place that researchers new to the field or even old to the field can go to and find the resources they need. So, sorry that was a whirlwind tour of stuff, but I hope that gives you a flavor of the progress that's being made and the amount of energy that's been directed toward KCNQ2. And I think that speaks volumes for the bright future for the condition. So thank you.
Has there ever been a clinical trial? And if not, I think the answer is no, but
Yes, there has.
if not, how close is that to happening?
Okay,
Does anybody
Scotty, yeah, the short answer is yes, there has been a clinical trial. Do you want to answer that, Scotty, or shall we just turn that over to Al? Let's let Al take that on.
I wasn't involved in the clinical trial, but I was aware of it. So the drug retigabine was approved many years ago for adults with focal onset epilepsy. And then it was tried, sort of ad hoc in patients with KCNQ2, but not as part of a clinical trial, just an observational experience. But then that drug got pulled from the market. It had a side effect that made people turn blue. Literally, their skin turned blue. And so people weren't using it. The company said, let's not waste any more money, pulled it off the market. A second drug company in Canada named Xenon had an interest in this drug. They reformulated it so that it was less prone to this chemical reaction that created the blue color, and they wanted to formulate it in a liquid form that could be given to small children so they could do a clinical trial in KCNQ2 related disorders. And they launched that trial, but the trial design, and this is something we learned, the trial design did not take into consideration all features of the disease. They wanted to simply see if the drug controlled seizures. And they said, but you can't be younger than 2 years old to be in the trial. So by making it so restrictive, they got 8 people to enroll in the trial, not enough. And they said, we can't do this. We're not getting enough people to enroll. That was a learning experience for everybody that told us, for at least this condition, counting seizures is not the endpoint that we need. So that clinical trial, unfortunately, didn't progress. Fortunately, there are now other molecules. 2 other companies have other molecules that will maybe act the same way, and they're trying to get those approved by an easier route, which is to test them in adults with focal onset epilepsy. So those trials are ongoing now. They're not going to be for children, but if they get the drug approved, there's an opportunity to then maybe do observational studies in children and eventually create the right clinical trial for children. So does that help your question?
And I'll add one more thing on that, because that was a great answer. When we talk about non-seizure clinical trial endpoints, what that actually means is, how many of you have children with some level of gut issues? How many of you have children who have speaking difficulties? How many of you have children with any sort of eye problems whatsoever? How many of you have children where learning is more difficult? Okay, those are non-seizure endpoints. How do we measure the things that make KCNQ2 DEE so hard for our kids? Figuring out ways to capture what we all know KCNQ2 DEE is, that's what we're trying to figure out. And so what we're doing is, the other piece of this is, we're getting much wiser about how we approach clinical trials. So when we have something that didn't work according to plan, we're now going, okay, how can we do this with our smart scientific team? How can we work with the government? And to be clear, all of this is regulated by the FDA, right? The Food and Drug Administration, you can't just say, hey, I'd like to do a clinical trial. Let's just do that. You've got to get approval for this. It's highly regulated. And of course, many of our population are children, and so that gets more difficult. So if you go back to when we started the foundation 10 years ago, and we didn't have a hope of any of this, to where we are today with drugs in the pipeline, I just want people to feel comfortable and confident that we are powering on and we have lots of irons in the fire. So maybe either Al or Brooke, are there, without naming names because you may not be able to, are there other clinical trial options that you see that might be coming along in the next couple of years, to answer the second part of that question? When's the next clinical trial you think we might have?
Yeah, so I think a KCNQ2 focused clinical trial is something that is sort of top priority for us. As you heard, we've got some really exciting medications that can do that I believe can do significant good in our population. And so those, while those medicines, the scientists are still doing their development, we can do our part to really get ourselves clinical trial ready, right? This is something you'll probably hear a lot about. So I'd like to be optimistic and think that we can, if we're ready as the drug is ready, like we could be in sort of this few to 5 year time frame, right? But that it is going to really, be boots on the ground. It's thinking about these non-seizure endpoints. It's working with groups like Inchstone to make measures that can actually record the change that we see. So I do believe there's going to be, a clinical trial focus on a KCNQ2 DEE in the very near future, hopefully even within the lifespan of the CZI grant, and so that I can guarantee I'm still here. But I also, as Sarah was mentioning, there are ongoing clinical trials for potassium channel activators happening now, just unfortunately not happening specifically for our community. And once those potassium channel activators are hopefully approved, they will be available. The last piece I'll add to that, because you will hear from some scientists today that are looking at other molecules like, again, sodium channel blockers or serotonin system modulators. They have trials in clinics which are called basket trials. So that means you don't have to have Dravet to get in the basket trial. You don't have to have Lennox-Gastaut syndrome to get in the basket trial. All you have to do is meet the criteria. Granted, those criteria right now, I believe, are mostly seizure-based. Those more close to the trials will tell you more about that. But for those of you where that is a feature in your life right now and that you might qualify, you will have the ability to participate in these clinical trials for the small molecules that are coming through clinic today. So you'll hear more about that later this afternoon.
I'm just going to see if we have, okay, we have another question over here. Yeah, we have 2 more questions. I'm going to say we'll do a rapid round. We'll do 3 more questions, and then we're going to take a break. And let's go for your question, please.
Yeah, so my understanding, and correct me if I'm wrong, is oxcarbazepine never had an FDA trial on KCNQ2 specifically. So I guess it's a 2-part question, actually. One is, are there other current medications that are already approved, could be repurposed for KCNQ2 that are being looked at? And 2, what are the trial process? What's the approval trial process in other countries? And is that also potentially another way to fast track some of this process?
Yeah, there's a little bit to unpack there, but I think that, yes, there's never been a clinical trial for KCNQ2 and sodium channel blocking drugs like carbamazepine, oxcarbazepine, and others. It's just become accepted that that's the drug of choice. And those drugs are now generic. So there's no financial incentive for a drug company to pay millions of dollars to run a clinical trial. But that said, the medical community has kind of accepted those, and that the existing evidence, clinical series and case series, is sufficient that people have accepted it. But yes, there's not been a clinical trial. And it's something we discussed in our scientific advisory board. Should we propose a sort of a kind of a test? Can we do a clinical trial without seizures as an endpoint? Hey, let's just use carbamazepine as an option. No one's going to pay for that. That's the sad thing. So other countries in their regulatory process, I'm not that well versed in that. You want to?
I can talk a little bit about that, and mostly from the dinner we had the other night with Yuri from Camp 4, and again, some experience I have in industry myself. But yeah, so different countries do have different regulatory structure. And so there can be advantages to going to other regions first. We were talking about Australia has become a very popular place, especially in genetic medicine, to start things, just the way they do things. It's sort of a bit more like scientist-based, like if you can get sort of scientist endorsement, their regulatory agencies kind of lean on their scientific teams to get things through. Europe is also another space in which clinical trials sometimes are more easily initiated than in the United States currently. So yes, we are not sort of rate limited by the FDA by any means. There's ways to start clinical trials other places, and they do all have sort of their own unique set of systems.
One last thing on that is that there are what I'm going to call cousin disorders, right? I always think of genetic epilepsies as an alphabet soup. We've got KCNQ2, there's SCN2A, SCN1A, CDKL5, Lennox-Gastaut, et cetera. There are trials happening, and you're going to hear about one this afternoon that's very, very interesting. And it's not us, but all of this work is collectively useful for what we're doing. Would you agree with that? Okay, all right, I saw another question over there, yes?
Thank you guys for the work you're doing. Quick question about carbamazepine. You had mentioned during the some of the trial research you're doing with the mice. You're seeing some changes not just with the seizures, but some of the neurological or some of those additional changes and some of those other endpoints. How realistic or optimistic are we that medications like this or others like it can provide some kind of solution for some of these other endpoints aside from seizures?
It's a really good question, and it wasn't my work. I highlighted some discussions yesterday. I think the mouse study was done by Laurent Billard, and they, they purposely looked at things other than seizures in the mouse, and they saw some improvements in certain behavioral traits that they could elicit in the mice. They also estimated cognitive function in the mice and saw that there was improvement, but it took longer than just stopping seizures. So you can stop the seizures pretty quickly, but to see this sort of benefit in the cognitive behavior of the mice, that took a longer time. And it brings up an age-old question, is the impaired development of the brain due to seizures? So controlling seizures early will improve the neurodevelopment long-term. I think that's still an unanswered question. I think people still think that it probably is not entirely all about seizures, that you can stop seizures very early and yet impaired neurodevelopment still manifests later. So, but that said, is carbamazepine disease modifying, which we would say different from just being treating symptoms? I think it's an open question, but there's some hints that it may be, or that class of drugs may have some disease modifying benefits. Not the whole story yet, but maybe.